Good Monday morning, everybody. Time to get back to work. So, we're going to begin on chapter 11. And in chapter 11, we're hitting the foundation of genetics and how we started to learn what all we do know about genetics. So, we begin with Gregor Mendel. Gregor Mendel, father of genetics, did his work in the 1860s and was largely ignored. People didn't pay much attention to his work because, face it, we didn't know anything at all about DNA, how heredity was passed on, and so they didn't take too much stock in everything that he found out. But he began his work when he was serving as an Austrian monk. He joined the monastery because he wanted to further his education. He was a very intelligent young man. His family was poor, and the only way to get a college education was through the church, Catholic Church, which pretty much founded college educations and further education. So he decided to join the monastery, and he studied mathematics and physics and was very good at those two things, and he became a teacher. And then later on, he became the abbot as he got older in the monastery. Now, older back then was like late 30s, early 40s. So he kind of went into a semi-retirement, and having grown up on a farm, he enjoyed farming, and he was very curious about looking at the plants and wondering how and why the traits got passed on from one generation to the next the way they did. He was one of the very first scientists to combine math with the observations that he was making as he grew his pea plants. And he's very well known for the meticulous manner in which he kept all of his records. He absolutely every cross, every seed that he planted, every plant that grew from those seeds, he recorded what he saw. And in the very first year of his work, that included 7,324 installments of what happened. So for every seed he planted in that first year, he recorded it. And he did it for 20 years. 20 years. He was very curious about how traits passed from one generation to the next. And what he didn't know was that in his observations, he perfectly described what happens in meiosis. And we'll see how that correlates later on in this chapter. His procedure that he used to study inheritance has been followed and used to study genetics ever since his work. For the longest time, they didn't have the tools that they needed to see what he had described, but once we did have the tools, we found that he had described everything exactly the way it happens. So his work, like I said before, was not given recognition until long after his death. Really wasn't until the 1900s, the mid 1900s, where everybody said, wow, he was kind of an amazing guy. So his work. Here's a lot of terms. Most of them are from this section, and I think you've already defined them. If not, you need to know what these terms mean. We have genetics, which is the study of inheritance, a trait, which is any one particular characteristic, and we're going to be talking about hybrids and hybridization, and that's where you take the different versions of a trait and you cross them. So you take two true breeding parents that have different versions of the same trait and you cross-pollinate them. Genes, those are segments of DNA that control the expression of traits. Alleles are the different versions, and what we mean by that is if we are looking at the flower color of a plant, that would be a trait. That would be a gene. And to hybridize, we would take and cross the different colors. So in pea plants, that would be a purple flower crossed with a white flower. And purple and white would be alleles. Mendel discovered the principle of dominance because in his work he found that in his crosses, one trait disappeared completely and then reappeared in the next generation. So he determined that one allele was stronger or more dominant than the other, so it could hide the other trait if they were combined. So he found that the trait that can do the hiding is dominant, the one that can be hidden is recessive. He also found the law of segregation, in that parental alleles are going to separate independently into gametes. You get two alleles for every trait. You get one from your mom, you get one from your dad. And when in meiosis, gametes are formed, those two, the one from mom and the one from dad, separate into separate cells, and that's called the law of segregation. We'll discuss that more later as well. Gametes we've talked about before, those are sex cells, so egg cells and sperm cells, or in the case of plants, we're looking at pollen. Pollen grains are like the sperm cells, and then there is, again, an ovum or an egg cell. Genotype is a word that you need to know. If we're asking for genotype, you're going to give me the genetic makeup of the organism, and to do that, you're going to give me the letters representing the alleles. You also need to know what phenotype is, and that's describing the physical characteristics. In other words, what does it look like? If the alleles are the same, we say it is homozygous. And if the alleles are the same and they're two capital letters, it would be homozygous dominant. If the alleles are the same and they're both lowercase letters, the alleles are homozygous recessive. If you get two different alleles, the capital and the lowercase of that letter, then the alleles are different, and our genotype is heterozygous. All of those terms you need to know. So make sure that you do whatever you need to do. A lot of you still like to do the flashcards. That's awesome. But make sure that you know what these terms are. I'm going to stop the PowerPoint now because I'll run out of time, and then we'll continue on a new video. So bear with me here for a few minutes and we'll get another video made.